find its origin and insertion, contract it, shorten it, see what the action is, and then figure out that if the muscles are on this side of the body, the antagonists are on the opposite side. It's opposite action. Muscles that work together are synergists. Muscles that work against each other are antagonists. So if you just know, if I've got a muscle here, then what's the antagonist? Look on the other side of the model. Okay, it's back here. It's very easy if you make logic of it. If you're sitting there trying to memorize it, forget it. It's very confusing. But if you can make sense of it, you'll do better. So I'm going to talk about how we approach that so you can get a head start on that, okay? And then that will be tonight. And then on Thursday, we're getting right into muscles, okay? All right, so let's go ahead and go through some of this. It might be a little bit longer, but I want to get through this part one because most of tonight's lab has to do with the information from this PowerPoint. Being able to identify the organization of a muscle, the skeletal muscle, as you saw on the diagram in your lab map, lab 16. Just need to be able to do that. Okay. So let's go ahead and get started on this. We're going to start with part one. This unit is broken up into 3 distinct units. Number one is just the organization. It's a lot of terminology. You thought it was bad the last time. It's worse this time. So if you need to write words down, you write them down, okay? And you keep saying it to yourself and to each other until you've got it, okay? So first part is the organization. Then the second part is, we're looking at, in the first part, the organization of a muscle as a cellular structure. Then we have to look at it as an organ, which is part 2. So how does it work together? How come we need to have ATP? Why do we have to have mitochondria? So we're going to talk about energetics. We're going to talk about muscles as a whole organ. And then we're going to finish out the unit talking about how all of this works together. Okay? So there's 3 major parts to this. And the handouts are broken down into that to help you. I would really strongly suggest, if you haven't done so already, get familiar with YouTube on sliding filament theory, neuromuscular junction, all the animations that are in that, Amoeba Sisters, what is that, Crash Course in AMP, just looking at, just put it into the search engine, sliding filament theory, and then get an animation, 3 minutes, and you see everything, how it all works together. Okay, so get used to doing that to kind of assure yourself of what you're thinking is happening is really happening. Okay? All right, so we have more than 600 muscles in the body, and we're going to learn only about 63 muscles. And these muscles have multiple ways in which they're named. Okay? So some of these are going to have, like, for example, the biceps brachii. It has 2 heads. Bi means 2. Ceps referring to head, like cephalic region, right? So that word root means the head. And then the brachium has to do with the arm. So the biceps brachii tells you it's got 2 heads and it's found on the arm. Okay? And you can see that some of these muscles have a tendinous connection. So they're going to attach indirectly onto bone. You need to know the name of the bone on which it attaches, not the landmark. So you don't need to go, radial tuberosity, brachioradialis, no, all you need to know is it's going to come down, it's going to go on my brachium, on my humerus, and it's going to come down onto my ulna, okay? That's what all you need to know. So if I say name and insertion, you give me the name of the bone. So we've got them, we kind of pulled out from all those landmarks. They're important landmarks. You're going to revisit them in the clinical approach when you start looking at patients and you're using those to identify structures under the skin. So it's going to help you there. Okay, so we've got then a number of different ways in which these muscles are named. Some muscles are going to be convergent. Pectoralis major. For example, it is the superficial most muscle on the anterior chest. So superficial most, and it has then all of this attachment right here. You can pretty well tell where it's going to go. Clavicle, sternum, okay? And and then it's going to have fibers that converge, and they're going to attach onto my crest of my greater tubercle, on my humerus. So they're going to come across, attach, and what's it going to do? All I have to do is shorten the muscle. I can tell what it's going to do. That's what you're going to do on the test. You don't have to memorize actions. If you know where the muscle is, just shorten it. You can feel it get thicker. That's why you know that you contracted it. You know what the action is. Some of our muscles are fusiform. So the biceps brachii has two, it's a spindle shape, okay? It's got two heads on it, two attachment sites. The less movable end is the origin. The more movable end is the insertion. Okay? So that would be fusiform. Then we have muscles that are parallel. So on the model, you'll see that it looks like a strap. Okay, kind of imagine. The sartorius has an origin, anterior superior iliac spine. That's where it starts. And then it's going to come on to the medial side of the tibia. So it's going to cross over two joints, my hip, my knee. So that muscle right there, when I contract it, it's a strap. It allows me to sit cross-legged. And the reason why it's called a sartorius, I don't know if it was called that or what, but sartorius, the word sartorio refers to a tailor. Okay? Person who sits in the old days cross-legged and will sit there and sew and alter clothing, right? So this allows you to do that. It allows you to then sit cross-legged when you're sitting in a chair. Okay, another shape would be then the circular. In this case, you really don't have an origin and insertion. There is a tendinous connection. For example, your eyelids are going to be circular muscles. Your lips, circular muscles, okay? So, got that shape. Then we have the multipennate, which means it's just got multiple parts to it that then are going to attach at a single point, okay? And then we have bipennate too. It almost looks like, the biceps brachii. This is erectus femoris. It's got two parts to it, okay, but it's one muscle. And it's in the anterior thigh, superficial most, and it's going to be part of your quads group. So I'm going to ask you, which of the following is not part of the quadriceps femoris? So you need to know the group name, because that's how you're going to talk about it when you're talking about it in the gym, when you're building up your quads, if you're doing some resistance training. Okay, and then we have the unipennate, which is then it's like a single fan coming down. And you can see then where these muscles are identified in the body, and you're going to have to tell which one's superficial most on the anterior chest, which is the superficial most in the upper back, which is the superficial most on the lower back, which is the superficial most in the gluteal region. So you need to know relationships of muscles. That's the only drawback of not doing cat dissection. There's not many other reasons why, The only thing that's good about doing cat dissection is you can see the layering effect. Most, when you see diagrams, you see them as static structures, but they're layers that are going to then pull the muscles together. Okay, so every muscle that you have in the body, can you believe, is its own individual organ. Has all four tissue types present. Remember, that's how we define an organ. It's got epithelial tissue, connective tissue, nervous tissue, and the muscle itself. So those would be the four components, principal tissue types that make up an organ. So every muscle is its own special organ. Now we can classify muscles according to the type of action or function that's associated with them. You're going to hear some terms, and you'll read them in your lab manual that says it's the prime mover. The prime mover is the most important muscle that does that action. So for example, if I'm looking at my deltoid, the deltoid is going to have an origin on my clavicle. It's going to come around on my scapula, go down the spine of my scapula, and then it's going to insert right here on my humerus, on my deltoid tuberosity. So when I shorten that whole thing, it's what gives me the shape to my shoulder. When I shorten it, it's going to do that. We identify that action as abduction. abduction. That was at the end of the last unit. So the prime mover for the abduction of the arm is going to be my deltoid muscle. Now, there are going to be other muscles that help it because muscles don't work in isolation. They work together collectively. So you're going to have then, along with the prime mover, synergists. So a synergist is another muscle that's in close proximity that's going to help do the same action, but it's not the primary one. Okay? Prime mover is going to be the primary one. The agonist is going to be it's just a different term for the term prime mover. Now, we already learned that one of the ways in which the body is going to stabilize joints, because that's the weakest point in the skeleton, right? Is at the articulations. We're going to have at least one muscle going over it. Okay? And so they're going to help to fix that and stabilize that particular joint. Okay? So you see here then, I've got then here's my anterior anatomical position, anterior muscle on my arm. That's going to be my biceps brachii. It has two heads to it. Okay? One is on the coracoid process. You don't need to know that. You just need to know it goes on the scapula. Okay? And then it's going to come down and it's going to insert onto my onto my ulna. Okay? So it's going to come down here. No, to my radius. Okay, so it's going to come down here, and this entire muscle, two parts of it, is going to be the prime flexor of the forearm. Okay, so if it crosses two joints, there's a joint right there, there's a joint right there. That means it's got two different sets of actions. So my biceps can do this, and it can do this. Right? Whenever a muscle crosses two joints, it's going to have the action on the distal bone. So wherever that might be, here is going to have an attachment here onto my radius. So it's going to help to flex my forearm, but it also has part of it directly onto the humerus itself. So that's going to help me do this. So both my arm and my forearm are going to then be moved by that biceps. Now this is on the anterior side. My antagonist is on the opposite side. So my triceps brachii is on the back of the arm. So if you get stuck and you go, the question is, name the muscle, give its origin, name the muscle, give its insertion, name the muscle, give an antagonist. Okay, all you need to do is to look on the opposite side of the appendage and then tell me the name of that muscle, and that's going to be your antagonist. So if you know where it's located, all you have to do is look on the other side, and we've got it. Okay, so we've got synergists and antagonists, origins, insertions, lots of terms. Okay? All right. So we've got three different kinds of muscles. Now, in this unit, we're going to spend the majority of time on skeletal muscle. I'm going to give you an overview of cardiac muscle, but for heaven's sake, you've got a whole chapter, a whole unit on cardiovascular. So you're going to go over that again on the cardiac muscle in 50B. And then smooth muscle is found along the walls of all my hollow organs. So that's urinary tract, respiratory, everywhere. You've got smooth muscle. In this one, we're going to spend the majority of time on my skeletal muscle. Okay? So then I've got three kinds of muscle. And tonight in lab, you're going to be looking at slides that show the three different types. You've got to be able to differentiate between the three. Tell me which muscle is in the field of view. Okay? And there's going to be some characteristic clues that are going to tell you which one it is. So a couple of items associated with the skeletal. Obviously, skeletal is attached onto the skeleton, which allows them for mobility to accomplish work. The cardiac is only associated with the walls of the heart. And then the smooth muscle is on the walls of my hollow organs. Now this is what you're going to see on the slides. You're going to see then on skeletal muscle, you see these striations. Now when you get to your 400 is where you're going to see these prominently. Okay? So when you look at that, you'll notice that this represents one muscle cell. Okay? And so you see multiple striations, and the nucleus is shoved, it's so filled with actin and myosin there's no room for the nucleus, so it gets shoved to the side. So that's one of the clues that you're looking at in skeletal muscle. On cardiac muscle, you'll see striations as well. Okay, so you'll see the stripes. But the characteristic clue is that on cardiac muscle, you see these very distinct bands, like right there, right there. Those are called intercalated discs. And so you'll have a slide with a pointer on the intercalated disc, and it will say, name the muscle in the field of view. When you see that intercalated disc, it's got to be cardiac. And this is actually a gap junction. It's how two cardiac muscle cells can communicate with each other in order to synchronize the pumping, okay? So their contraction. The other thing about cardiac muscles that makes them unique, striations is not unique, because if there's striations over here and striations here, that doesn't make it unique. The intercalated discs is unique. Unique is something that's only found in that type, okay? So the other characteristic is branching. So you see this cardiac muscle, it has they have tendencies to branch. That way you can send out multiple messages through multiple branches to get the whole unit to contract as one. So that's a characteristic of cardiac muscle, unique. So I can say intercalated disc, and I can say that it's branched. Now, skeletal muscle is under voluntary control. I got to send the message from my brain down to the muscle to contract. These other two are under involuntary. I don't have to think about it. The heart's going to beat on a uniform rate unless I under the influence of adrenaline or other kinds of drugs and, you know, hormones, etc. So I can then alter that, but I don't control it. It's just a result of interaction with various chemicals. And then here, this is under involuntary control. I don't have to think about, I just chewed my food, swallowed the food, down in my stomach, gonna digest my proteins down there, then I'm gonna send it to the, you know, we don't have to think about that. It does it automatically. So these two are under involuntary control. Okay? Okay. The other couple of things. This is a unique feature of skeletal muscle. It's multinucleated. During embryogenesis, muscles are developing so fast that they don't undergo cytokinesis. So they become multinucleated. They don't separate out completely. And you can then have a multinucleated structure. And because it's multinucleated, we don't typically refer to it as a cell. Technically, it is a cell, but we call it a myofiber or a muscle fiber, okay? So we have then multinucleated, that is unique, okay, to skeletal muscle. The other, let's see, make certain, on my cardiac, I may have a couple of cells that have two nuclei, but no more. The most of them are uninucleated, okay? And then smooth muscle is always one nucleus. That's it, okay? And they have a tendency to be fusiform. So they look like they're tapered. Looks like a long, kind of a big tube that's tapered at the ends. And then they all line up together to form this kind of a sheet of muscle. And that then helps to form the wall. So there are no striations. It's under involuntary control. We will talk a little bit about smooth muscle and a little bit about cardiac muscle before the end of this unit, but just very briefly, just to kind of give you an idea of some pretty obvious differences between skeletal to the other two. Now we're going to learn a whole bunch of new words again, like I said, terminology, and you'll see that there are some prefixes that are associated with skeletal or for muscle. The word myo, muscle. Mes, like mesium, epimesium, okay? That's going to then be muscle. And then sarco, sarcoplasmic reticulum, SR instead of endoplasmic reticulum. So we've got then some terms that are just referring to muscle itself, okay? All right, so let's look at some of those terms. All right, the first thing I want to talk about are the layers. Now, this has to do with the organization. Some of you have already looked at this in the lab manual, first part of lab 16, and you can see then the organization of a muscle. This is an entire muscle, and then if we break it down into smaller units and smaller units and smaller units, then we're going to see the way in which that muscle is going to be organized in terms of contraction. When I contract a muscle, there's a difference between picking up a 1 pound bag of sugar versus a 25-pound weight. That should make sense to you. If I'm picking up a 1 pound bag of sugar, I don't need to contract my entire muscle and waste energy to do all of that. However, if I'm picking up 25% or 25 pounds, I need to now recruit more parts of the muscle to lift that 25 pounds. Doesn't that make sense? Yeah. So the way that we do that is that it's going to then activate certain parts of the muscle. Small part for small weight. As we increase the amount of weight, a greater portion of the muscle is going to be activated. Greater amount of ATP is going to be necessary to do the contraction. Okay, so we have then the most complex organization associated with this, with skeletal muscle. And you already found that out in that lab. Okay, and what we're going to see on each one of these, I'm going to go through the different mesium. Those are the connective tissue layers. Okay, and we're going to find that all three types of muscle have around each of the muscle cells a layer of connective tissue. That layer is called an endomesium. So if I'm looking at a cell, every cell has a cell membrane, doesn't it? Yeah. In a muscle cell, no matter which one you're looking at, that membrane is called the sarcolemma. We give it a special name. In reality, when we go into cell biology and you see then it's a plasma membrane, sometimes the old way is plasmalemma. When we're looking at a muscle cell, the membrane around a muscle cell, same configuration, phospholipid bilayer, but because it's associated with a muscle cell, we call it a sarcolemma. So superficial to the sarcolemma, okay, each one of these represents superficial to the sarcolemma, I have this layer of connective tissue called the endomesium. That's what holds the cells together. Okay, so the connective tissue that holds the cells together as a unit in the muscle is called the endomesium. All three muscle types have endomesium. So surrounding any muscle fiber, muscle cell, I'm going to have endomesium. Now in skeletal, I have a much more complex organization. So not only do I have an endomesium around each one of these represent the cross-section of each one of my myofibers, I have them bundled together. There's a bundle, there's a bundle, there's a bundle, there's a bundle. Each bundle, called a fascicle, is now going to be surrounded by connective tissue called perimesium. So the perimesium bundles the muscle cells into groups, okay? And then surrounding the entire muscle and continuous then with the tendon to attach onto the periosteum is my epimesium. So my skeletal muscle has these three layers. It has an endomesium around each of the myofibers, a perimesium which bundles the fibers together into a fascicle, and then holding everything together as one unique muscle would then be my epimesium. Okay. Each one of these is a different connective tissue. So I'm going to find some in my epimesium, dense, regular, on the outermost, and then dense irregular. Paramecium, dense irregular, and areolar, and then endomecium, areolar, and reticular. So all those connective tissues that we talked about way back when in Unit 2, we then have those now compounded and holding these the muscle together. Okay, so a couple of other things that we need to know. So this is, you have this PowerPoint, so it makes it very easy. Just kind of look at it. Okay, that makes sense. Under voluntary control, I have to tell my muscles, skeletal muscles, to contract. And then they can go very quickly or very slowly. If I'm doing resistance training, I do not want to, if I'm trying to bulk up, I'm not going to go like this, right? I'm bringing it on up slowly, and I'm going to bring it back down slowly under strict control, right? So I can either contract quickly or slowly. I can regulate how fast I'm going to contract. Now, if I go fast, I'm going to have a tendency to fatigue. So skeletal muscle fatigues. You know that when you're working out, after a while, your muscles I can't do it again. Done, okay? So they fatigue. So the quicker they are to contract, the quicker they are to fatigue. Cardiac muscle, no, we do not want fatigue. We want it to remain smooth and regulated and consistent. 72 beats per minute, okay? We want that to continue the entire time. Now, there are going to be times when I have to pump more blood to my legs to run away from the rabid dog, but the bottom line is that those are isolated events. In normal, I want about 72 beats per minute. Unless I'm really working out, I'm going to increase it, but then I got to get back down to resting phase. I don't want it to stop or to be overworked. And then the slowest of all of these muscles is smooth muscle. I mean, you eat dinner, it takes about 5 hours later, depending upon what you have in that dinner, 5 hours later to have that food completely digested down. 5 hours. To move it down from your stomach to your small intestine, to your large intestine, that kind of good stuff. So you want that to go very slowly because it's going to be very purposeful in terms of moving that food in that particular lumen. Okay? Okay, so we talked about the speed, okay? So think about, just make sense of it. Really, don't try to memorize these factoids. Try to make sense. Yeah, I need my heart to beat 72 beats per minute all the time. I need to slow down the digestion, move it along slowly. Okay, smooth muscle. As I said, smooth muscle, they are fusiform. They're held together by way of endomecium, okay? And they form these wide, long sheets. So if I'm looking through a cross-section through my colon, I'm going to find that there are 2 layers of muscles, smooth muscles, around the wall of that hollow tube. The innermost layer are circular. Okay, so we're going to find then a circular layer, okay, that we're going to find here. On here, superficial to that is going to be my longitudinal. So if I've got a tube, okay, I've got a circular layer first, and what it's going to do is squeeze, squeeze, squeeze to move the food through. The other one, the outer one, is going to shorten, shorten, shorten to move the food down along that tube. So I usually have 2 layers in the kidneys. I mean, not in the kidneys. Let's say in the urinary bladder. 2 layers. The innermost layer is going to be my circular, and then superficial to that, my longitudinal. The only one exception is in my stomach when I have 3 layers. I have an inner circular, superficial to that, longitudinal, and then superficial to that, oblique. So your stomach has a tendency to do this, to squeeze the food down, shorten so that it moves it along, and then it twists it too, like wringing a hand towel, okay? So it's got 3 different activities as it moves the food through, so you've got 3 different layers. Most everything has got 2. So smooth muscle is oriented that way, okay? 2 layers. So the question on the test will be, The superficialmost layer of the smooth muscle in a digestive tract is going to be my longitudinal layer. That's going to be true. Okay? So you need to know the orientation for that.
Question.
Uh-huh.
For the stomach, are there any oblique muscle?
Which one?
For the stomach, is it oblique?
Oblique. Yeah. It's kind of on a diagonal. Okay. Okay. All right, and then the cardiac muscle is the unusual most in that it's going to be oriented in bundles. Okay. So you've got then the way in which they're bundled and they're kind of organized is the way in which I got then a four-chambered pump. The upper two working together, the lower two working together, the atria versus the ventricle, and then I've got a left side and the right side. So I then have it organized in such a fashion to be able to then contract in a specific way to either take the blood from the heart to the systemic circulation to the entire body or up to my lungs. It's got two ways, two routes in which that blood is going to go when it leaves the heart. So I've got the muscle bundles arranged so that they can actually move them in synchrony. Again, intercalated discs. Big term, you need to know it. Okay, we did that way back when we first talked about muscle as one of the four principal tissue types, so it shouldn't be a new term. But now that it's related to muscle, you have a better understanding. It's a gap junction, allows for intracellular communication so that the entire unit contracts as single. You'll find out that there is a pacemaker, an SA node and AB node that are going to help control that. But for right now, you just need to know, hey, there's intracellular communication. That's going to happen. Okay? Again, autonomic neural controls that are going to regulate the pace. So on a regular 72 beats per minute, if your SA node is working all by itself, you might get 45 beats per minute. If you go down to the AB node, it's less than that. So you've got then kind of a built-in pump control, okay, regulation, control, contraction. Okay, so skeletal muscle is under voluntary control, and then these are long, elongated cells, multinucleated, and then cardiac is found only in the heart, smooth muscle is fusiform. Easy enough. All right, so let's look at a little bit of the organization on this. I'm going to go about another 15 minutes because I want to get through the material so that you can work in the lab. Okay. So, skeletal muscle is under voluntary control, and it's attached onto then the periosteum, a bone. So there's two ways in which skeletal muscle can attach. It can either attach directly onto the bone or indirectly by way of a tendon or an aponeurosis. Okay, so I've got two ways. Now, if I look at this, you can see that the outermost covering here is going to be my epimysium, and it's continuous with my tendon. We know what kind of connective tissue is in a tendon, hopefully, we remember. Dense, regular connective tissue. So the outer layer of the epimysium is dense, regular connective tissue because it's got to be the same as its connection. Then deeper to that, this is my dense, irregular. Okay, so you've got two layers that are in that location. Right, so that's my outer epimysium. As we said, depending upon which muscles that we're working with, they can fatigue very quickly if we have quick contractions that are associated with it. The quicker the contraction, the more, the greater the ability of fatigue. There are going to be other controls in there too, but for right now, we're just making that simple kind of an overview. It allows for body mobility, and that's what the primary function of skeletal muscle would be. And because we contract it, it contributes to 60% of your body temperature. Okay, so it really contributes significantly to your overall body temperature. They're extremely adaptable. You've heard of the term muscle memory, right? Okay, when you're learning how to play a piece on a piano, okay, you practice so frequently that you don't have to look at the music anymore after a while, and the muscles are moving along the way that they should because you've already imparted into them a muscle memory. Okay, so they are adaptable. And depending upon what we want to accomplish with them, I can make them work really hard, or I can make them work very weakly. So if I'm looking at then my biceps brachii and I'm lifting 1 pound of sugar versus 25-pound weight, I can adjust what I need to do. I mentally look at the weight and it says 25 pounds on the side, so you kind of go, okay, I'm going to have to put a lot of effort into this one here, okay? So you're recruiting then the muscle fibers to get the best contraction that you can. When you see the 1 pound box of sugar, you kind of go, oh, I don't need to put as much pressure on those muscles as I would with this 1 pound versus 25 pounds. So I can vary the amount of work that's associated with skeletal muscle. Skeletal muscle has 4 functional characteristics that are part and parcel. First one, excitability. They have to be able to respond to a stimulus. So that is, the information is coming from the nervous system and telling it to contract. And depending upon what the initial stimulus is, it could be quick contraction or slow. It's got to be excitable. It's got to be irritable. It's got to be responsive to stimuli. That's one characteristic. So if it has then a supply, an innervation, it's got to have some kind of a nerve ending that goes to the muscle to direct it to do something. Okay, so that allows for that excitability. It's got to be able to contract. Okay, that's called contractility, and it should be able to then be stretched or extended beyond its normal length. But then it's got to recoil and go back to its original shape. That's elasticity. So those are my 4 characteristics that are associated with muscle, skeletal muscle. It's got to be responsive to stimuli. It's got to be able to contract. It's going to have to be able to stretch beyond what its normal length would be if we needed it to. And then it's got to recoil back to its original shape in order to do the work again. Okay? So those are my 4 functional characteristics. So it's responsible for all locomotion. We already know that cardiac muscle is responsible for blood circulation, whether it's going to the rest of the body or to the lungs. It still needs to be the pump that's going to take the blood to that location. And then smooth muscle takes care of everything else. Okay, everything else, blood pressure, blood vessel diameter, the way in which you digest food, the way in which your urinary bladder functions, all of that is reliant on then the functionality of smooth muscle. Okay? Okay, so skeletal muscles are organs. So skeletal muscles are actually organs. It's got all 4 major principal tissue types present there. Okay? And then every muscle fiber. Now we're going to use the term either myofiber, I use the old-fashioned term, myofiber, or muscle fiber. That's equal to muscle cell. But because it's multinucleated and because it's got an unusual configuration, we don't usually call it a cell. We call it a myofiber. Okay? And being elongated makes it also this characteristic of a muscle fiber. It's got to be supplied with a nerve ending. So here is a cross-section of my spinal cord. And then coming off of that spinal cord, you're going to have these neurons, these motor neurons that are going to actually go in and innervate each of the myofibers. Every myofiber has 1 nerve ending. Okay, that's going to control it. So I can have a nerve ending, a motor nerve. We call it motor because we're moving a muscle. So the motor nerve, okay, the motor neuron can either go to just 1 or multiple or hundreds. It can branch. So I can have then, coming from a single nerve, multiple myofibers that are going to be innervated at the same time, activated at the same time. Just imagine if I had to do hundreds of them and each one had to have their own separate signal. Better to have one and branch it and get it all done at one shot, right? Makes more logical sense in terms of energy need. So we then have these motor units. We'll talk about that in a moment. But you can see that every one of these myofibers has a nerve ending. That's the important point, okay? And these fibers are going to be very close to and surrounded by blood capillaries. I need, in order for a muscle to contract, I gotta have oxygen. Contraction is reliant on ATP. We're gonna have to review glycolytic pathway, Krebs cycle, electron transport one more time in this unit. We know that the mitochondria require oxygen to make ATP. So I have to provide then that oxygen for the muscles to contract. In addition, I have to supply nutrients. Sugar, fatty acids, various nutrients in order for the muscles to contract. So you're gonna have a lot of capillaries surrounding that muscle. And you can tell if you ever look, you look at like meats, different kinds of meats. What do you suppose the red color comes from? Like blood capillaries. That's why, you know, beef is red because it's got lots of blood vessels in there, right? And it's got a different flavor than maybe chicken. Chicken still has blood vessels, but not as abundant as in beef. That's why it's got a different coloration. Have you ever noticed the difference between white fish versus red fish? Red fish like salmon, white fish like cod. Cod's white. Salmon is pink. It's reliant on the amount of myoglobin and capillaries that are found in that muscle. So the more abundant the blood vessel, the more redness associated with that particular kind of muscle that you're eating. So dark meat versus light meat in a chicken. Looking at a thigh. Well, the chicken's got to run. They got to run away from the foxes, and they got to run. So they're going to have lots of blood vessels in their thighs, but do they use their wings to fly? No. That's why wings are light, right? And do they need to use their pectoralis muscles? No, because they don't fly. So breast meat is going to be then light. Thighs and legs, they gotta run. Got dark. Lots more blood vessels. So it's all related to the amount and the abundance of blood vessels and myoglobin, ultimately, okay? So we're gonna have variations, and we're going to talk about that because you need to know the difference between those kinds of muscles. Okay, again, 3 layers of connective tissue. The innermost layer, this one's going backwards. I usually go from the outside in, but I'll go with the PowerPoint just to make certain it's consistent as you study. So around each of the myofibers. So here's my muscle, the whole entire muscle. It's wrapped with a connective tissue layer continuous with the periosteum. If I continue to go into the smaller, this represents one cell. That's one myofiber. That one myofiber is held together in a bundle, like a pack of sticks. In between each one of those cells is connective tissue to anchor them together. That connective tissue that we find in there is called endomysium. Now I have a model that you're going to be using tonight in lab that shows a myofiber. That's what that model is. It's a myofiber, and you'll see that around that entire unit it looks kind of like a fibrous network. That's connective tissue. Remember fibers? So we've got then fibers, and that's going to be my endomysium. So the endomysium is on the outside of that model. Okay, then, continuing on, surrounding each one of the bundles, each bundle is called a fascicle. So it's wrapped around, like a saran wrap around it. That's called a perimysium. Okay, and then these bundles are held together as a single unit by way of an epimysium. So the epimysium is the outermost dense regular connective tissue. That's continuous then with the periosteum of bone. But it's continuous actually with the tendon, dense regular connective tissue, which is then going to be part of the periosteum, dense regular connective tissue, so that it's all continuous and holding then the muscle onto the bone. This is what it's going to look like on cross section on a slide. So on the outermost, you can see that this is a cross section, and all these little round circles represent myofibers. So I have then surrounding the entire connective tissue is my epimysium. Here's a fascicle, here's a fascicle. So surrounding each of those fascicles is perimysium. Inside is my myofiber, and in between each one of those is my endomysium. So if I had a slide out there, you can tell me which kind of connective tissue is at the tip of the pointer. Okay, you'll have it big enough to be able to then differentiate between and tell what the different connective tissues are. Two ways in which muscles attach onto the bone. Talked a little bit about this in passing, direct versus indirect. Direct means that it's directly attached onto the bone itself. You know that deltoid tuberosity on the humerus? That's direct connection of deltoid to the bone. Then I have my biceps, which is then tendinous connection, which is then going to be then indirect. Now, most often we think of this tendon as my connection, but there are going to be places in my body, if I look at my abdominals, there's no bone here, okay? All I have then is an aponeurosis. So I've got a tendinous as a sheet onto which the muscles attach. So that's called my abdominal aponeurosis. Okay. So a tendon and an aponeurosis are the same. It's just that the tendon is cord, and then aponeurosis is a long, flat sheet. You will have to be able to identify those little cross, the for part of the six-pack, etc. Okay. I'm not going to do that one. I want to get to my here. Okay. So I have then looked at a muscle. Again, just to make certain that we understand every muscle is an organ. How do we define an organ? All four connective tissues are all four principal tissue types are present. So I can find an epimysium, perimysium, and then an endomysium. Okay, so we saw this as a unit. Here's a fascicle. So let's take one of these and then highlight it. So in this fascicle surrounding that is my perimysium. Okay, that's my dense, irregular connective tissue. And then within that fascicle are my myofibers. These are my individual cells. Okay, so if I look at then a muscle fiber, this is what it's going to look like. Now what's missing from here is my endomysium. They wanted to pull it away and just show you this is the outer edge of a cell. That membrane is called a sarcolemma. So if I have it on my myofiber and I said name that structure, it's going to be a sarcolemma. Or name the structure, that could be endomysium, because you'll see the two on there. And so around the sarcolemma is going to surround the entire cell. It's a cell. However, it's a modified cell. Doesn't look like your typical animal cell. It's got very different structures that make it a muscle fiber. So if I look through, do a cross section through my muscle fiber, I will see that there are bundles of myofilaments. Each myofilament, each bundle is called a myofibril. Now this is where the myos get all messed up. Myofiber equals muscle cell. Myofibril is a bundle of myofilaments. Myofilaments, we're going to find out, are actin and myosin. Okay? So what a cell really is then is just a wrapped bundle of myofibrils, each consisting of actin and myosin, and these myofibrils are arranged in such a way that you see this striation. Dark, light, dark, light. So this dark light that you see here has a name. So I have a model which illustrates the various components of a myofiber. Okay? And you can see then the A bands, the I bands. We're going to go into that detail in a moment. So this then is my muscle fiber. I've got a model of that. So I've got a list, and it says up at the top, myofiber. Don't tell me that's a muscle on a lab test. It kind of looks like it, but it's really one myofiber. And we're looking at then this and all the structures. So you can see on here, these nuclei shoving along the side, they're kind of pinched out in space because they're so large. And then in between the myofibrils, mitochondria. Got to be able to contract the muscle, and they require then ATP. So I've got then my myofiber, which is striated. Okay? Outermost, sarcolemma. And then that's equal to cell membrane. Then if I look on cross-section, this is just a highlight. Here's my myofibril, myofibrils. And those are actually bundles of myofilaments. Okay? And the way in which they are arranged, they show a very specific cycle of dark, light, dark, light, dark, light banding. And that's due to the presence of the myofilaments, how they're all in register. So my dark bands are called A bands. Those are the dark. The light bands are I bands. And some people go, well, what the heck is that? Why do we call those A bands and I bands? Isotropic means that light passes through. So my light bands are I bands. Isotropic. Light passes through. That's why the I. And isotropic, without light. That's my dark band. So my dark band is my A band, and my light band is my I band. So you can see that register of dark, light, light. And that's due to the myofilaments that are in the myofibril. Okay, so each myofiber can be as long as a third of a meter. That's about a foot. Meter, well, third. Yeah. 1 meter is like 1 yard. Okay, so actually, yeah, 1 foot. Okay, so then I have then varying diameters. I can have lots of fibers in there or few fibers in there. So they can be variable. If I'm looking at my fingers, the muscles in between my fingers, there's going to have small numbers. If I look at my quads, I'm going to have lots. I'm going to have a lot. They're going to be bigger because it's a bigger muscle. The smaller the muscle, does that make sense? Smaller the muscle, the less the myofilaments. Okay. Just, again, try to make sense of it. Okay, now if I'm looking at a cell, what was the fluid matrix in a cell called? Oh, wait. So cytoplasm, wasn't it, in the cell? Now we call it sarcoplasm. It's equivalent to muscle cytoplasm, okay? So the sarcoplasm is where I'm going to have then, it's like the cytosol, it's where I'm going to have my organelles. So that's where I'm going to have then a variety of organelles, such as my mitochondria, my glycosomes. I've got a new set of organelles that I'm going to find in the sarcoplasm that make it unique to other types of cells. Glycosomes. Glycosomes are bodies that will take glucose and polymerize them into glycogen. Remember the polysaccharide that is stored in humans is glycogen? 2 places in the body. Skeletal muscle, liver. That's it. When those storage areas are filled, it then gets dumped into an adipocyte and gets converted into a lipid. So I've got glycosomes. Why? In order for a muscle to contract, I have to have ATP. I have to have ready glycogen to be right there just to make more ATP. And then I have myoglobin. Now look at that word, myoglobin. It kind of looks like hemoglobin. So hemoglobin is the protein that we find in red blood cells that carry oxygen. In muscle cells, it's called myoglobin because it can give up oxygen faster. So if I'm contracting muscle, I need to get the oxygen that I brought in there, release it quicker to make ATP energy. So I'm going to have myoglobin. And it's the myoglobin that contributes to the coloration of muscle. Okay? So again, two unique structures that we find in a myofiber. Glycosomes, the word some always means body, glyco, glycogen. Myoglobin, globin refers to the heme, like carrying oxygen, but it's in muscle. Okay. We have then in my muscle fiber, I'm going to have myofibrils. Myofibrils are organelles. They're bundles of actin and myosin, but they work together collectively as organelles. And I can have a variety of numbers. The more of the myofibrils I have, the greater the strength of contraction. So if I'm trying to build up my quads, I'm going to have a lot more in my quads than I'm going to have in my biceps. Okay, it's a bigger muscle group. Now here's then the detail. Okay, so myofibrils. Okay, this myofibril is going to look like this. This is what it looks like diagrammatically. That's what you worked on in the first part of the lab. Okay, and you can see that there is a difference in color, and it's intentional. The thicker red brick ones are myosin filaments, and the smaller ones are actin. So my two myofilaments are actin and myosin. Now I'm gonna correct this up on this. I'm gonna say that the two proteins in the myofibril are actin and myosin. I can say that. The thin filament is actin and its associated proteins, and the myosin is the thick filament is myosin. Okay? So some of you, when you studied that, you saw that the actin had troponin and tropomyosin. Actin, troponin, tropomyosin equals thin filament. Does that make sense? The protein itself is actin. The troponin and the tropomyosin are regulatory molecules on my actin. That whole unit is called a thin filament. And then my myosin is the thick filament because it's got globular heads. Okay. So I've got then my thick and thin filaments. Now you can see then that between everywhere where I see the myosin, which is the brick red, and the actin, the combination of the two makes it really dense. Light can't go through it, so it's called an A band. A band is equal to actin plus myosin. The I band, if you look at it right here, the only thing that's in an I band is my thin filament, right? So my I band is actin and, well, the thin filament, and then my A band is thick and thin. You'll see that on this diagram that this kind of you can call it a Z disc, I call it a Z line, it doesn't really matter. Okay, so the Z disc serves as the attachment point of my actin. So from Z line to Z line, that unit right there is called a sarcomere. So the question on the lecture test would be, which components would you find in a sarcomere? Or what structures are the outer boundary of the sarcomere? Okay? Yeah, question?
So would it be appropriate to say a subunit of the myofilament is a sarcomere?
The subunit of the
Myofilament is a sarcomere,
I wouldn't say that. I would say it is a functional unit of the myofibril. Okay. So it's a functional unit. That's where all my contraction occurs. Okay, I'm going to give you a break here, and I'm going to finish this out in lab. And I'm going to use my models to help illustrate it. Okay.